Unit 4 Chemistry, Chapter 15, Part 3. Third type of organic molecule here, biomolecule, is the proteins. Now, proteins are made up of amino acids. Amino acids have got an amine group and an acid group. Okay? They also have in the middle one carbon, one hydrogen, and a variable group called a Z group. And look, if you did biology, you will probably be used to calling this an R group. Please don't call it an R group. In chemistry, an R group contains only carbons and hydrogens. These groups, these Z groups, contain very often other elements such as sulfur, oxygen, nitrogen. So you must use the Z. Okay? You may also see these called 2 amino acids or alpha amino acids sometimes. So, this is the general structure of it. Our carboxylic acid group, our amine group, and our Z group sticking up from that carbon, the second carbon. There's a big variety of Z groups, which you're not going to learn. You're going to look at in your data book. But they confer specific properties on them. So they might be an acid group, if they've got a carboxyl group in there. They might have an amine group, which will make them alkali. They might have a group they might be cysteine, which has a sulfur in it, which can make a disulfide bridge, which is a very strong covalent bond. Okay? Because they affect the bonds the amino acid can make, that affects the protein structure and what's made from them. Let's think about the amino acids on their own now. We're going to think first of all about the common part. So all amino acids have got one amine group and one carboxyl group. So the amine group can act as a base, the carboxyl group can act as an acid, okay? Which means they're amphiprotic. They can accept and donate protons at the same time. So if you take an amino acid and you put it in solution, you're going to make something called a zwitterion. In that zwitterion, the carboxyl group has given away a proton, and at the same time, the amine group has accepted a proton. Okay? So what we're going to have here is a doubly charged ion with a negative on one side and a positive on the other. Okay? This also means that it can act as a buffer though, because if I put that amino acid in solution and then I add too many hydrogens, well then this group here is going to be able to accept another hydrogen. This is going to stay as it was, but this will go back to being COOH. So the whole thing becomes positively charged. In an alkali solution, the opposite thing happens. This group here can give up another proton. This will stay negatively charged, so we will end up with a negatively charged ion. And because we can accept and donate both, we can act as a buffer, which means we're going to stop the pH of the solution changing by as much as it would otherwise. Now, those common parts are also what's involved in reacting to link two amino acids together. And when we link them together, we make an amide link. Again, sometimes it's called a peptide link, but just as with the glycosidic, it's better to use the term of the functional group, because anytime you see it, you know it will definitely be an amide link. Okay? If we link two together, we make a dipeptide. If we link multiple ones together, we make a polypeptide. The order in which we link our amino acids together, that's called our primary level of protein structure. Okay? So here we have a carboxylic acid and an amine group, okay, which can link together, and yes, they're attached to the rest of it, but we can make our amide group. Okay? So this would be our primary structure of this segment, and notice it is a segment. We put open bonds on both ends. Anytime you're asked to draw a segment, you must have open bonds on both ends. And this primary structure goes valine, serine, cysteine, alanine. And it will keep on going. There'll be many more amino acids in there. Once we've got that primary structure, we've got all our chain of amino acids linked together, we can then link it together to make hydrogen bonds, make our secondary structure. And it's these bits here that do it. Okay? So it's the oxygen in the C double bond O and the hydrogen on the NH, which are going to attract each other. Okay? So let's imagine this one was here, and then we had another chain and it looped round, and you had a C double bond O sticking up here. In between there and there, that's where you're going to get a hydrogen bond holding different amino acids of the same chain together. Our Z groups, these variable bits, are not involved yet. So this is our secondary protein structure. And we've got two shapes. We can either make a helix or a pleated sheet. Okay? When we then move on to the tertiary structure, this is where our Z groups get involved. So we look at our different Z groups and we say, okay, well, if we've got OH and NH bonds, well, then we're going to have a polar bond, so we could have a hydrogen bond. If we've got a carboxyl group, that can donate a proton, so it's going to become negatively charged, and an amine can become positively charged. Two charged things attracting each other, we've got an ionic bond. If we've got cysteine, and it's only cysteine that can do it, that's going to drop the hydrogen in its Z group, and it's going to make a covalent bond between two sulfurs. Or if they're non-polar, we're going to get dispersion forces, which you see here labelled as hydrophobic interactions, but because we're chemists, we know that that's actually a dispersion force. Some proteins stop at the tertiary level. Most stop at the tertiary level. But some go another level where we've got two different chains of polypeptides linked together. And that's called the Quaternary level of structure. The type of bond that holds it together is exactly the same as what holds together tertiary level. Okay? So this is hemoglobin, which has two alpha chains and two beta chains. So it's actually 4 different polypeptides held together. And here we can see lots and lots of alpha helices in play. Now, just as with the fatty acids, there are some amino acids that we can synthesize, and there are some that have to be eaten. The ones that we have to eat are our essential ones. So these are your essential amino acids. These ones are completely non-essential, you can make them. Conditionally non-essential means as long as you've got the right other things, the right bases, then you can make those. But it depends what you've got in your diet.